Hi, my name is Steve Traynelis. I'm a professor at the Department of Pharmacology and Chemical Biology here at Emory University School of Medicine. I've been working on glutamate receptors for over 30 years, 25 of those years here in Atlanta. I'm also the director of the Center for Functional Evaluation of Rare Variants. And those two entities do an enormous amount of work on research topics that are relevant to rare diseases, particularly the GRIN, the GRIA, GRIC, and GRID variants. So I'm standing right here in what looks like kind of a messy set of equipment. We call these recording stations, and this is just an example of some of the innovative work we're doing to try to advance an understanding of these variants. These are two automated robotic, what we call patch clamp systems, built with our partners and friends down at Georgia Tech, where we will be able to not just slowly evaluate glutamate receptors, but automate that and increase the speed tenfold. The equipment and the software, as well as very simple everyday pieces, have been redesigned, re-engineered, and our goal is to increase our ability to look at data 10 or 100-fold. And this is what's going to be needed to evaluate how all the many variants that we're learning about in our patient population actually impact function. We also do quite a bit of work to try to understand in a native system, in actual neurons, what the variants will do. What I showed you before is a system where we make DNA in a test tube, and we trick a cell into making that so we can study it with great precision. But of course the neurons are really different than the cells we work on. And so we have other systems that look similar to the one I showed you, but this one is geared up for recording from native neurons and microcircuits, and so it allows us to begin to ask questions about how those variants can impact circuit function. And of course we need to know that if we're going to try to figure out how to mitigate any problems they would have for the circuit, and ultimately develop, think of, validate new approaches to therapy. These neuronal recordings also allow us to take advantage of genetic manipulations. We can introduce into these neurons variants with gene editing with CRISPR techniques, but also transfecting them with DNA. And then we can go and we can evaluate, once the neuron has shown a deficit that we introduced, we can test various ways to genetically revert that and show that it's working. So we have that going on as well. So in the other room I was showing you apparatus that we use to record from small cells, mammalian cells, neurons, but this much simpler system is a real workhorse preparation, and this is a oocyte recording system. It's also automated and set up, and we've actually got, and here's just one of them, but we have 6 or 7 of these dual systems running in the lab. They can all run automatically. And this is a system where we also characterize the effect of the variants, but importantly, it's set up with a lot of automated valves, and we can go through a significant number of FDA-approved drugs looking for compounds that are already approved and can be used in patients that might, in some cases, reverse the effects of the variant and provide relief for some of the symptoms. The pharmacology won't be a perfect cure, but it's an important stopgap measure, and in some cases will be a major improvement to quality of life. And so we're able to do that with the oocytes. We have another little simple system here where we can inject those, and between this and, as I mentioned, here's the second one here, and there's a whole bunch of them down in another room, we're actually able to get through a large number of what we call pharmacology studies and evaluate existing medications. And so I think the last thing I want to tell you in closing is, of course, I'm taking this video today at the end of the day where people are home, but you should all realize that we have a large number of folks all committed to trying to come up with cures and ways to improve the lives of the families and the patients with these GRIN variants. So you're not alone. A lot of people are on your side, and we're working hard.